There are several different analytical techniques that we can use to study DNA and RNA, and many, if not all of these, are used in molecular biology labs today in different ways. So first of all, we can use absorption of UV light. We know that nucleic acids absorb UV light at 260 nanometers, as we discussed previously, and protein can be absorbed at 280 nanometers. So if you have a particular sample and you want to see how much DNA you have, how much RNA you have, how much protein you have, well, it's a little difficult to distinguish between the DNA and RNA. You absolutely can determine the differences between the nucleic acids and proteins. And the standard way to do this is to use a UV spectrometer, and you can actually measure the numbers at where it's being absorbed at these different wavelengths of light. You can also take advantage of the fact that DNA can be sedimented after centrifugation. So all of these different nucleic acids, proteins, other parts of a cell, the mitochondria, the nucleus, all these things, they all have a different weight to them. So if you perform centrifugation, you can specifically isolate whatever you want to study. One of the common ways that this sedimentation is used is through cesium chloride purification, and that's one of the things I used a lot when I was a graduate student. So when you have cesium chloride and your mixture of DNA, when you spin it at very, very high speeds, we're talking about like 40,000 revolutions per minute, you can get a very clear, distinct band of your DNA. And you can get several other bands of other things that you may or may not want. You can just use a needle, you can go in, you can pull out that particular band of the DNA. And it'd be very, 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 very pure DNA from this process. You can also take advantage of the denaturation and renaturation capabilities of nucleic acids. So what we're looking at here is melting temperature. If you know the melting temperature and the length of a particular piece of DNA, and by melting temperature, I'm talking about the temperature at which the two strands of DNA are no longer together through the hydrogen bonds, but the hydrogen bonds get broken and you have two single-stranded pieces of DNA. If you know how long that piece of DNA is, you can roughly predict the percentage of GC content just based on that melting temperature. And the reason for this is because if you remember, between the A's and the T's, there are two hydrogen bonds. And if you look at the structure of the bond between the G's and the C's, there are three hydrogen bonds. So areas that have more A's and T's, they're going to melt or come apart at a lower temperature than areas that have a lot of G's and C's. And you can actually correlate that to the GC content within that piece of DNA. Some more techniques we can use, molecular hybridization. So anything that is complementary to your DNA or RNA can bind to it. And so one of the ways that this is used is you can use a probe that is radio-labeled. With a phosphorus group, you can radio-label that with P32. It will bind specifically to whatever region you make that specific to. You can go in, you can design it, we can order them, you can buy all the materials that you need, design these probes, and it will anneal specific or bind specifically to that region of DNA. We'll talk more in a minute about gel electrophoresis, but you can separate them out based on size, and you can know that this is going to be very, very specific binding. You can take this one step further. Instead of using radio labeling, you can use a fluorescent tag on the probe instead. And this is called fluorescence in situ hybridization, or FISH for short. Similar process, just a little bit more specialized in the molecular hybridization. Because it's a fluorescent tag, you can literally tag this while it's still in the cell. And so, for example, you could follow the DNA, you can look at the structure, you can look to see if the ends are broken, just by using these different tags. You can use reassociation kinetics and repetitive DNA. So similar to before, we talked about the melting temperature and the reannealing process. If an area has very, very highly repetitive versus if it's very, very diverse, you can see how quickly that they will anneal. And this will provide information on the size and complexity of genomic DNA from a particular organism. The last technique we'll talk about right now is gel electrophoresis. In gel electrophoresis, we take advantage of the fact that DNA is negatively charged. It is negatively charged because of that phosphate group on the backbone of DNA. So, if you place DNA in an agarose gel, there's usually wells at the top, and there's usually an electric current that you can apply. One of the ends is going to be a negative charge, one of the ends is going to be a positive charge. We have the negative charge where the DNA is, the positive charge at the other end, because when you apply the current, the negatively charged DNA is going to be attracted to the positive end and it's going to try to migrate or travel towards this end. However, there are gel particles in their way. So small pieces, they can get through this process pretty quickly, pretty easily. They'll migrate faster. Pieces that are larger, they're more likely to get stuck. They're not going to travel quite as quickly. And so we know that the molecules move at a rate that's inversely proportional to their length. The smaller they are, the faster they'll move through the gel. After this, you can then incubate with Ethidium Bromide. Ethidium Bromide is a stain that will allow you to put UV light through your sample, and it puts a kink in the backbone of the DNA, and so it will literally glow if you look at it under a UV light. You can do some other techniques. You can incubate with different fluorescent dyes, but the end result is the same in that the smaller fragments will travel further down through the gel, larger fragments will be not traveled as far through the gel. You may see different intensity in bands. This is usually proportional to the amount that you have. So if a band is darker like you see here, you may have just simply more of it than if you see less of it like you do in this band or this band here. So concluding week 5, what I want you to do for your homework this week is 2 parts. The first part is to do the Exploring Genomics BLAST part in your book. This is going to be on page 264 in your book. And just go ahead and follow the directions as written. For the week 5 homework part 2, just complete the Mastering Genetics homework as you have previously. Of course, there's a week 5 quiz, and please participate in the discussion forums as you've done previously. One other thing I would like you to do this week is to look through and at least attempt to read one of the historical papers that I linked on D2L. You can read through the insights and solutions on these papers at the end of chapter 10. The work from these papers is what we described in the textbook, or is what is described in the textbook. What I want you to think about is what similarities, what differences do you see from how that work is described in the textbook to the original paper?